Good day, Homo sapiens. How are you? And welcome back to studying chapter 2 of this Prescott's Microbiology book. And last time we were on the staining and staining types, and we only studied how to prepare our specimen, like how to fix it on the slide, and what mistake we should avoid, and what are the methods, the dyes and simple staining, right? Like what kind, like how does the staining actually works. And they are saying that the general staining are of basic dyes and acidic dyes, like it is based on the charge. Like basic dyes have positively charged groups, so it attaches to the negatively charged compounds on the cell or inside the cell, while the acidic dyes have negative group, for example carboxylic group or OH group, so it has negative charge, and so it attaches to the positively charged cell structure. And this was I think we were done till here, and let's see what else there will be. And can I go and check first if the live is working? Let's go. Wait, give me 20 seconds or 10-15 seconds. Yeah, it's working. Yeah, I just needed to see if it will. Yeah, if it was working. Thank goodness it is working. And let's go back without any delay. So they are saying that the staining effectiveness of ionizable dyes, guys, the above two, the acid dyes and the basic dyes, were ionizable dyes because they become in a charged form, right? May be altered by the pH. So its effectiveness can be changed because of the change in the pH, since the nature and degree of the charge on the cell components change with the pH, right? We know that because of pH means the concentration of 10, right? Negative log. So thus acid dyes stain best under acidic conditions when proteins and many other molecules carries a positive charge. Basic dyes are most effective at higher pHs. Right. So the higher pH would mean 10 raised to power minus 11. If it is 10 raised to power, or if you can say if the hydrogen concentration, the pH is 2, that would mean that the hydrogen concentration is in minus, right? 10 raised to power minus numbers. So that means the hydrogen concentration will be in lower quantity. That means the positive charge will be in low quantity, and they are saying that the basic dyes work effectively because basic dyes are negative, so like there will be no interference of these basic dyes and the cell that cell that is positively charged. So basic dyes are positive. What am I saying? Basic dyes are positive, so there will be no interference because of the pH, higher pH. Oh, I wanna beat my body. So higher pH means lower hydrogen concentration. Yeah, I was kind of right that basic dyes are plus, right? Plus. So if we have low plus quantities, low plus charges, that won't interfere with the positive charge of the basic dyes, and that means more basic dye will attach to these negative cell structures, right? Instead of this hydrogen ion disturbing the or all concentration. The same opposite case happens with the acidic dyes. Dyes that bind to the covalent bonds or because of their solubility characteristics are also useful. So this is what we were talking about, the ionizable dyes, right? Now we are talking about the dyes that form covalent bond, not ionic bond. So, or because of their solubility characteristics, they are saying that are useful as well. How? For instance, DNA can be stained with the Fulgen procedure in which staining compound shifts reagent is covalently attached to its deoxyribose sugar. Sudan 3, Sudan black selectively stains lipid because it is lipid soluble but will not dissolve in aqueous portion of the cell. So I didn't get a single word. They are saying that DNA can be stained by the covalent dyes, right? For example, by the Fulgen procedure. It's a procedure by which DNA is stained by these covalent bond forming dyes. And here what happens? A staining compound, the Schiff's reagent, attached to the alright, deoxyribose sugar. Of course, oxygen has sugar and it has deoxyribose, means oxygen is in the middle of the sugar, you can say double bond O. So it is covalently attached to the Schiff's reagent. I don't know what the Schiff's reagent looks like, so let's go and see what it is. What does it looks like? Schiff's reagent. Guys, here is a solution that detects the presence of aldehydes in a substance. It is used in histology and microscopy to identify mucous substances, glycoproteins, and other substances. Alright, so it is for aldehyde groups. Why does it attaches to? Wait, deoxy doesn't mean it has the aldehyde group, right? Attached to deoxy. What am I writing? What kind of stupid questions are these? Can we see here somewhere? Was my question not clear enough? At this times the is used to rejection of aldehyde reacts with the aldehyde group to form characteristic magenta or purple color. In DNA, the aldehyde group is especially found at the reducing end of the sugar in the backbone, which is the deoxyribose sugar. Deoxyribose sugar DNA has a hydroxyl group, we know, at its 3 position, right? When the H at 2 position, hence the deoxy in the name. Oh, okay. With exposed aldehyde on the sugar ring which can participate. Oh, I must be dumb. Yeah, okay, so it is the aldehyde group in the deoxyribose sugar. So they are saying that the attachment there happens with the covalent bonding. Well, the Sudan 3 or Sudan black. Alright, the Schiff reagent gives a magenta color, right? Well, the Sudan 3 I think gives a black color. Selectively stains lipids because it is lipid soluble but will not dissolve in aqueous portion of the cell. So they are saying that it will only stain lipids in the cell membrane, right? The lipid bilayer, you can say. So it selectively stain only lipids because it is lipid soluble. We don't have to go into the chemistry of why it is lipid soluble or how it is structured. I think we are, personally I would like to go in detail with that. Microorganisms often can be substained very satisfactorily by simple staining in which a single dye is used, right? They are saying usually by simple staining or one dye is enough for staining microorganisms. Simple staining value lies in its simplicity and ease of use. Like just dump the microorganism a slide and pick it out and it will stain. It's as simple as that, right? So one covers the fixed smear with a stain for a period of time, washes the excess stain off with water, and blots the slide dry. Yeah, simple as that. You make a fixed smear, then stain it or put a stain and leave it for a short period of time, then wash the excess stain with the water and let this slide dry with the stain. Wait, what happened? I am so sick of these ads. Alright, so basic dyes like crystal violet, methylene blue, carbofuchsin are frequently used in simple staining to determine the size, shape, and arrangement of the prokaryotic cells. I think these crystal violet, methylene blue, and the kind of things we already studied, and they said that they are known as what salts? Acid salts? Here, look. Yeah, chloride salt. Sorry for my mistake. Yeah, chloride salt. So methylene blue and fuchsin crystal violet, these are basic dyes, means they are positively charged, and they bind to the negatively charged. Guys, here is the big boss of the staining, and that is the differential staining, right? And differential staining have 2-3 more categories in it, but even in the differential stain, the gram staining is way popular, way too much popular in microbiology. Like if you are dealing with a bacteria, go straight towards the gram staining. It will give you idea if the bacteria is gram positive or negative, and then it drastically reduce the bacteria that you have to go through to identify the microorganism that you want to see, right? Because its structure is different, and you can't see under the microscope without staining if it has double lipid bilayer and the peptidoglycan, because the gram positive bacteria has thicker layer, you can say it has less layer, or you can say it doesn't have the lipid bilayer, a second lipid bilayer, right? Lipopolysaccharide layer. Unlike gram negative, like it is caked. Its polypeptide chain is caked between lipopolysaccharides, so it is more resistant to staining than the gram positive one. So that's why the cell wall is so drastically different. So you can say half the bacteria between the, or divide the bacteria between the gram positive and gram negative. So it has gram staining technique, acid fast staining. So there are 2. I think there will be more as well. Right. So these 2 are more popular, or you can say more important, more basic. The gram stain developed in 1884. This is so far back, by the Danish physician Christian Gram. Yeah, I remember. Easy to remember its history because of its name similarity with, not similarity, it's the exact name taken from the scientist's name. Is the most widely employed staining methods in the bacteriology. Exactly, bacteriology specifically. Alright, we are not talking about the fungi and these kind of things. So it is an example of differential staining procedures that are used to distinguish organisms based on their staining properties. Differential staining definition is: these are the procedures that are used to distinguish organisms based on their staining properties, right? Every organism, when takes stain, its internal structures are different from others. Some has more quantity in them than others. Some has more bigger shape. Some has smaller shape. Some are rounder. Some are thinner. Some are thicker. Like a lot of characteristics appears when you stain them. So use of gram staining divides the bacteria into 2 classes, gram negative and gram positive. Like I said, it's because of the cell wall. And the negative staining is set. Wait, here they talked about negative gram staining, where the background but not the cell is stained, right? So this is not the negative we are talking about here, because I made a great dumb mistake in my previous life to talk about random about the gram negative ones, but it wasn't that. So that's negative staining, and this gram negative is different. This gram negative refers to the cell walls, while that negative staining refers to the background staining as well. You can say the background is colored and the cell is not colored, so it is negative. Usually the positive staining is that we stain the cell, right? That's the normal process. That's why it's positive. But when we do the opposite thing, we stain the background and not the cell, that becomes the gram negative. But in this case, gram positive and gram negative bacteria both are stained positively. Both stains go towards the cell and not the background. But the only thing is, the gram positive ones retain the color when we wash them with alcohol, but when the gram negative bacteria are washed with alcohol, or you can say ethanol, I think, so it removes its lipid layer, and with that it removes its stain as well, so it appears transparent as well again. So we have to use a secondary stain, double staining we do, and this is not the simple staining, because we do use the second stain, that is safranin, pinkish color. So it appears. So gram negative has pink color, gram positive has violet color, purple color, and both the stains. Gram positive stain is only the crystal violet, while the gram staining is first stained with the crystal violet, and after washing it is again color stained with the safranin. So the gram staining procedure is illustrated. Wait, why was I talking about the procedure when it's all written here? Dang. In the first step, the smear is stained with basic dyes crystal violet, the primary stain, right? First is color with the crystal violet, and that is the basic dye. This is followed by the treatment with iodine solution functioning as mordant. Using iodine solution, we kind of fix that stain, right? Like it acts as a glue, you can say act as a glue. Wait, we can still, mordant meaning. Yeah, is a fixative that combines with the dye or stain to intensify or improve its color fastness. Fastness means, drying, you can say attachment. So the iodine increasing the interaction between the cell and the dye so that the cell is stained more strongly. The smear is next decolorized by washing it with ethanol or acetone. Yeah. What's decolorize means we wash it. Like we wanna rip apart that stain of it if it is going to be ripped apart later. So if it is not removed by the ethanol or acetone, mean it is stained perfectly. So this step generates a differential aspect of gram staining. So now we are going towards the step in which we can explain why the gram staining comes under the category of differential staining, and it's because of this. The gram positive bacteria retain the crystal violet, whereas the gram negative bacteria lose their crystal violet color and become colorless after washing with ethanol or acetone. Finally, the smear is counter stained with a simple, that's the term, the counter stain. First is the primary stain, the second stain we use is the counter stain. It's again the basic dye, different in color from the crystal violet. And that is safranin. The most common counter stain colors the gram negative bacteria pink to red, and so the range is more than pink. It can lead to the darker color red, and leaves the gram positive bacteria dark purple, right? Like you don't know when you are coloring the smear, like which color it retains, right? So you just do processes step by step with you can say 2-minute gap. So you just do every step, right? You don't know even till the end if it is gram positive or if it is gram negative, right? After washing, you know that whether like you treat both whatever comes after washing, and you put the safranin, and after treatment with safranin, if it remains purple, that means it is the gram positive, and if it is changed to the pink, like it is removed colorless, so of course it's gonna take the pink color as well. So again, they said that figures 2.14c and 2.15b, 2.14c. So this is 1.3, 2.13, this is 2.14. Here are the gram stains. Look at this. The purple colors, the purple bacilli are the gram stain or gram positive, and this is the crystal violet color, while these one, the gram negative, are in pink color. They call it red, but to my eyes it looks like a pink colony, right? And they talked about B as well, methylene blue stain of the methylene blue. Methylene blue is a simple stain, guys. They are talking about a simple staining in which we don't need to counter stain it, and it's not just for, I don't think it's specific to just gram staining. So of Coryn bacterium or Coryn bacterium, I don't know what to say the scientific pronunciation. I hope they have a dictionary. Alright, so these are the steps. Let's look at this. Number 1, crystal violet primary stain, cell stain purple. Right, when you put this crystal violet primary stain, cell stain purple, all of cells come in purple, whether gram positive or gram negative. Step 2, you put iodine, cell remains purple because it just fastened, or you can say it advanced the adherence of the stain, so nothing changes. But step 3, when you put the alcohol decolorizer, you want to remove that stain. So there where the difference comes from. The gram positive cell remains purple because they don't lose their stain, while the gram negative cells become colorless. Like, look at this. Can you see the difference between these? You will be saying that they both look similar, so they must have the similar cell walls, but no. This is gram positive and that one is the gram negative. When you put saffron in the counter stain, gram positive cell remains purple, while the gram negative cells appear red. Means pink. Why do they use the word red? I don't get it. And this is how you see, look, purple. This is the coccus bacteria, cocci, I think. Coccus is singular, cocci is plural, or am I talking the opposite way? And these are the bacilli, no, these are the bacillus, right? They are singular. Bacilli, what's wrong with my brain? Coccus versus cocci. Coccus, oh yeah, coccus singular, cocci plural. What was it again? Guys, sometimes, these kinds of thing, you suddenly don't know which one's positive and which one's plural and which one's singular. Wait, if it's coccus is singular, that means bacillus singular as well. Okay, well, we get it. Look, they took the diagram from here. Maybe not. They just look similar because they are all bacteria. Alright, wait, is there something written here? Steps in the gram staining procedure. Result from the gram stain. The gram positive cell purple are Staphylococcus aureus. The gram negative cell reddish pink are Escherichia coli. Alright, so this is the real picture, okay? Real life bacteria that are the picture taken from the microscope. So they are saying the purple cells are the Staphylococcus aureus. Guys, look deep, look strongly at this picture. These are the Staphylococcus aureus, which you will be coming across more because they are common. Gram-negative bacteria, Escherichia coli, of course. Simple Bacillus Escherichia family bacteria. Well, this one's E. coli. Alright, so acid phase staining is another important differential staining procedure. It is most commonly used to identify Mycobacterium tuberculosis and Mycobacterium leprae. These are both the same genus, but the species is different. And this is also the differential stain. And how does it do the differential part? Now let's dive deep into this one. The pathogens responsible for tuberculosis and leprosy respectively. These bacteria have cell walls with high lipid content, in particular mycolic acid, a group of branched-chain hydroxylipids, which prevents dyes from readily binding to these cells. So I think it's the hydroxyl part of the lipids that might be interfering with, attachment of the stains. Or and they are okay. So till now they are just saying that both the lepra and the tuberculosis cell wall has high lipid density, high lipid amount, and both are specifically, you can say, mycolic acid, this type of lipid, the mycolic one, and it is the branch chain hydroxylipids. They are saying that this mycolic acid prevents the dyes from binding to the cells. However, Mycobacterium tuberculosis lepri can be stained by harsh procedures. If you are not going to be a good stain, then we are going to use rough procedures, such as Gill-Nielsen method, which uses heat and phenol to drive basic fusion into the cell. Gill-Nielsen method, which uses heat and phenol. Alright, so the phenol is a chemical and heat is like working as iodine to fix it, to drive basic fusion into the cell. Basic fusion, again, it is the basic dye and not the acid dye. Just as that one was the Fulgen procedure in the covalent bond forming dyes, this one is the general procedure for acid phase staining, in which we use this Gill-Nielsen method. In this, phenol and heat is used, and the dye that is used is basic fusion, and it is used to drive that dye inside the cell. Once basic fusion has penetrated, Mycobacterium tuberculosis and Mycobacterium lepri are not easily decolorized by acidified alcohol. Again, we do the washing process for this, but the dye is so penetrated inside the cell that it is not easy even for the alcohol to bring out that dye, or and thus are said to be acid fast. Alright. Acid fast. So there, this fast means fixed, kind of. Or you can say acid alcohol, acidified alcohol. What is acidified alcohol, guys? What is the difference between alcohol? I didn't notice that they use this kind of word. Difference between acidified and alcohol and alcohol. Like, I know they put acid in the alcohol, right? And alcohol is the hydroxyl group, yeah. They are saying the most common is the ethanol. So acidified alcohol is what? Acidic alcohol refers to alcohol solution in which an acid, usually a weak acid like hydrochloric acid or acetic acid, has been added. The acid lowers the pH of the alcohol solution, making it slightly acidic. Oh, lowers the pH. So the general alcohol is not that acidic. Oh, of course, because it has OH group. How is it going to be acidic? Alright, so it makes it more acidic. So that's why usually alcohol is not acidic, but when you put an acid, weak acid in it. Wait, alcohol is often neutral solvent with no significant acid or basic properties. Okay, well, even though it has OH group in it, doesn't mean it is leaning towards the alkaline. It is more towards the neutral, but when you put a weak acid, it automatically changes into acidified alcohol and are said to be acid fast. Wait, I know what acid fast means, but I need to be more concise about shut up. Excuse me. Characteristic of a certain bacteria. Wait, don't tell me we use acid fast just for the Mycobacterium. This is ridiculous. We produce, or you can say we made a whole technique for just a single type of Mycobacterium. resistance of a certain bacteria to being colored by acidic solutions due to their reaction helps. Alright, so I think neither the normal alcohol can decolorize it, or neither the acidic alcohol, so they are said to be acid fast, means they are acid resistant. Non-acid fast bacteria are decolorized by acid alcohol and thus are stained blue by methane blue counter stain. Wait a second. Okay, so I thought they were differentiating between tuberculosis and lepori, but in reality they were differentiating between tuberculosis, lepori, and the other bacteria. So when you put this stain, the what stain was here that was used? Phenol, not phenol, basic fission is used. When you use this dye for all of the bacteria you have in the culture, so the staining penetrates into the cell of only the lepori and the Mycobacterium tuberculosis, and they are not easily decolorized then. But the other bacteria, they don't have that acid fast ability, and they are decolorized. And so when you put another stain, for example here they are saying methylene blue counter stain, so they retain that methylene blue color instead of that acid fast staining color. So let's go down and look at the image 2.14 here. Acid fast stain, red cells are acid fast, blue cells are non-acid, because that is methylene blue. Well, this one is fuchsin. Alright, so there is nothing more into that. I was just checking if there was any other diagram. By the way, I am curious about if there are any other differential staining. What are all the types of differential staining? Forget my, or forgive my spellings because I don't care about that. So gram staining, they didn't have to explain. Don't explain to me. Just put the names, guys. Just put the names. Primary stain, carbofusion stains bacteria red. Oh, so we did what? Wait. Don't tell me they used the carbofusion word. Carbofusion. Where is the basic fusion? Yeah, I was sure that I didn't read the word carbofusion. Oh. Alright, so right games are staining for blood smear and parasitic infection. What the heck? All of these staining are differential staining, guys. What the heck? So let's leave it at that. I think we are okay with these two. So staining specific structures. This kind of differentiate between the organelles, right? Like this is the differentiation inside the cell, or between the cell structures. Many special staining procedures have been developed to study the specific structures with a light microscope. One of the simplest is capsule staining, because there are bacteria that you can't color their cell wall or cell membrane unless you pass through that capsule. So why not stain the capsule? A technique that reveals the presence of capsules, a network usually made of polysaccharides. This makes it easier for us to say if the bacteria is capsulated or not. And so we can focus on identifying the bacteria through the capsule category, the capsule list. We have the capsule bacteria list. We have, instead of going towards all the bacteria, right? So it narrows our range of work to identify the bacteria. So a network usually made of polysaccharides that surrounds many bacteria and some fungi. Again, it's a polysaccharide and nothing else. Cells are mixed with the India ink and the nigrosin dye and spread out in a thin film on the slide. So wait, is it the same? Is it the same dye or the different dye? Same dye or different dye? India ink or nigrosin dye? Wait, nigrosin dye is India ink. Is India ink same as what? I forgot the name. Nigrosin, okay. Nigrosin. Look at them. They will. Oh, okay. So the chemical structure is same. It's just the brand naming that will make it different. Okay, so that's why they use the or word. And spread out on a thin film on the slide. After air drying, the cells appear as lighter bodies in the midst of the blue-black background because ink and the dye particles cannot penetrate either the cell or its capsule. Okay, so this is the staining of the background. This is negative staining. Right. They have explained. Thus, capsule staining is an example of negative staining because the background is colored while the cell is not colored. When the background is colored, the cell appears more prominent on the background because it's no longer transparent versus transparent. The extent of the light region is determined by the size of the capsule and of the cell itself. Right, if you are going to see the structures within that background, it makes it possible for us to understand the size of the bacteria that we are dealing with. Although I don't think we will know how much is the diameter of the capsule and how much is the diameter of the cell. Yeah. So there is a little distortion of the cell shape and the cell can be counter-stained for even great visibility. Right. So you have stained the background and now you can counter-stain the cell as well. Yeah, they are talking about the cell counter stain, but we didn't even color the cell with the primary stain. Are they talking about the background itself? Components external to the cell. Alright, so they are saying we will explain the cell wall capsule and these things in chapter 3. And I'm looking forward to it. Endospore staining. It is important. These guys makes way too much problem for, dangerous diseases. Or you can say persistent diseases. Endospore staining, like acid-fast staining, also requires harsh treatment to drive the dye into a target because the capsule is strong. It is made for the external condition. It can resist almost every high temperature or high conditions to a certain limit. So we have to go beyond that limit for us to stain them. In this case, an endospore. An endospore is an exceptionally resistant structure produced by some bacterial genera, for example, Bacillus or Clostridium. Before going into that, let's look at the India ink capsule staining. So India ink, we did say that it is the negative staining. So the background is darkly colored. When we look at the cells, also the capsule does looks like something different than the cell wall. So the capsule is quite strong. It's more in quantity than the cell itself. So this is the Cryptococcus neoformans. So the endospore staining remains. So endospore species were bacteria and the not bacteria. What am I saying? Bacillus and the Clostridium. It is capable of surviving for longer periods in an unfavorable environment and is called endospore because it develops within the parent bacterial cell. Yeah, we know what endospores are. Endospore morphology and location vary within a species and often are variable in identification. Yeah, because endospores are different for every bacteria. An endospore may be spherical or elliptical or either smaller. This makes me so angry. The ads. Elliptical are either smaller or larger than the diameter